Hi, I'm Dr. Bill Carlazon, Principal Editor of NPP Digital Psychiatry and Neuroscience. Welcome to the DPN Deep Dive, our journal's AI-generated podcast that summarizes DPN articles in short, accessible episodes. Each episode is created using Google's Notebook LM, refined by our tech-savvy social media team, and approved by the authors to ensure clarity, accuracy, and approachability. Let's dive into this episode to explore the latest research in digital psychiatry and neuroscience.
We know that cognitive difficulties can have such a profound impact, right? Especially when we talk about serious conditions like psychosis. It often feels like this really personal puzzle, trying to understand and treat these challenges. Everyone's different. So today we're going to take a deep dive into some really interesting research. It's using cutting-edge brain imaging, actually, trying to get a better handle on predicting cognitive function, specifically in early psychosis.
Yeah, exactly. And the focus of our deep dive is this significant article. It's titled Predicting Cognitive Functioning in Early Psychosis: Factors Supporting and Limiting Generalizability of Connectome-Based Models. And this work, it's important to note, comes from researchers at Massachusetts General Hospital and Harvard Medical School. Really top-notch stuff.
Absolutely. So our mission today, really, is to unpack how these researchers used these detailed brain scans to find patterns in brain connectivity. And crucially, what does that tell us about understanding individual differences and maybe even improving predictions, for cognitive abilities in people going through early psychosis? So let's get into it. The core problem here, cognitive impairment is just such a difficult part of psychotic disorders, isn't it? It really affects daily functioning. And the big hope, I guess, with precision psychiatry is to find reliable ways to predict these outcomes for each individual person.
That's exactly it. And what's really fascinating here is how they're doing it. They use something called connectome-based predictive models, CPMs for short. Think of a connectome as like your brain's unique wiring diagram, its internal highway system. So these models, they essentially map out those highways using functional MRI data, fMRI, which shows brain activity, to find specific patterns, sort of like traffic patterns, and how brain regions talk to each other, patterns that might predict cognitive function.
Okay, so they're mapping the communication pathways.
Right. And they use data from the Human Connectome Project for early psychosis. Quite a large group, 93 participants. And then, really importantly, they tested their models on a completely separate group, 20 patients from Massachusetts General Hospital. That's key for checking if the findings hold up.
That independent validation sounds crucial, rigorous approach. So after all that complex modeling, what cognitive abilities could they actually forecast?
Well, they found these models could predict general cognition, sort of overall thinking skills, and also fluid cognition. Fluid cognition, that's our ability to tackle new problems, reason abstractly, think on our feet. The model showed moderate accuracy when tested on that independent group, which is, what's a promising sign for generalizability, though it did seem sensitive to exactly how much data, how many features they fed into the model.
Moderate accuracy, okay. But they didn't just stop at if they could predict, right? They dug deeper into how it worked, like which brain networks were doing the heavy lifting.
Exactly. They did this clever thing called virtual lesioning. Basically, in the computer model, they digitally switched off different brain connections to see which ones were most critical for the prediction to work. If you knock out a connection and the prediction fails, that connection is probably important.
Makes sense. What did that show?
It highlighted connections involving several key networks. The default mode network, that's the one often active when we're reflecting or mind-wandering. Also the somatomotor network, linked to movement and sensation, and the retrosplenial network, which plays a role in memory and navigation. And the frontoparietal network, really key for executive functions like planning and decision-making, that also showed up as important.
Okay, so several big networks involved. But there was a catch, wasn't there? Even with these sophisticated models, they didn't work perfectly for everyone. It sounds like they hit a bit of a wall with certain individuals.
That's right. And this is maybe one of the most interesting parts. They found the models consistently struggled for a subset of patients. And when they looked closer, parental socioeconomic status, SES, and how long someone had been taking antipsychotic medication seemed to be key factors.
Wait, SES and medication duration? How does that link to the brain model failing?
Well, the thinking is that the models weren't just learning pure brain biology. They might have inadvertently picked up on sort of stereotypical associations between these external factors and cognitive ability that exist in the training data. So if someone didn't fit that expected pattern, maybe someone with higher SES but unexpectedly lower cognitive scores, or vice versa, the model got confused. It suggests the model's predictions were partially influenced by these broader life context factors, not solely the connectome data.
Wow, okay. That's a really critical finding for precision medicine, isn't it? It really drives home that you can't just look at the brain scan in isolation. You need the whole picture, the person's context.
Precisely. It highlights the complexity.
And I assume, like all research, there were some limitations they acknowledged.
Oh, absolutely. For instance, the main data set, the Human Connectome Project for Early Psychosis, actually pooled data from 3 different research sites. Now, they did try hard to standardize things, but there could still be subtle site-specific differences influencing the results, though they did check for this, and the predictions still held up reasonably well, even after accounting for site. Also, some potentially useful information, like very precise education levels or exact medication dosages, wasn't available for everyone, which limited what they could include in the analysis.
Right, missing data is always a challenge.
And, while these CPM models are great for understanding the brain features involved, it's possible other machine learning algorithms might eventually achieve even higher raw prediction scores. So there's still room for improvement on the prediction front itself. But taking it all together, this research really underscores a critical point: trying to understand and predict cognitive outcomes in early psychosis. Well, it's definitely not a one-size-fits-all situation. It really seems to be about this complex interplay between brain networks and these really important individual factors like socioeconomic background, like medication history.
Absolutely. It feels like the findings strongly suggest that if we can integrate these broader clinical details, these sociodemographic factors, into our predictive models alongside the brain data, we could get much closer to truly accurate, personalized predictions. And that's not just about building fancier models, right? It's really about paving the way for support and interventions that are genuinely tailored to each individual's needs and context.
Exactly. Which leads us to perhaps a final thought for you, our listener, to mull over. How might these truly personalized predictive models, the ones that manage to account for both the brain biology and the complexities of someone's life context, how might they really transform the way we approach care? And how could they empower individuals who are facing these cognitive challenges in the future?
Something important to think about. Thanks for listening to the DPN Deep Dive. Until next time.